who will be talking about some past work and then into some of his new work, looking at how in vitro systems can be used to model cortical disorders. Thanks for joining us, Nico.
So thank you so much for this invitation. I'm very proud to be part of this seminar series. I'm Nicola Micali, associate research scientist in Pascura Case Lab, Yale University. And today I'm going to talk about how to model the early origins of cortical disorders using human neural stem cells in vitro. This is a collaboration with a team of Gabriel Samper from Hospital del Mar Research Institute in Barcelona, Spain, Carlo Colantoni, and also an assistant team. So if we talk about cortical disorders, of course, we need to introduce how corticogenesis works, right? So here in this slide, you see the early mouse telencephalon, and we can recognize regions of these early stage, which are called brain organizers. Organizers are secrete morphogens which instruct neural stem cells identity and pattern the different areas of the brain. So here we can recognize the rostral partnering center in the anteroventral domain, secreting FGFs. And then in the dorsal posterior area, we have the cortical hem secreting BMP Wnt, the anti-M in the lateral area, and the ventral organizer secreting sonic agent. So this cartoon shows how the neural stem cells progress across corticogenesis, right? Neural stem cells are not always the same across across development. So at the beginning, there are neuroepithelial stem cells, and then they give rise to radial cells, which form the neurons. And then radial cells transit across different states during corticogenesis, eventually becoming gliogenic. So disruption of corticogenesis may lead to cortical malformation and or neurodevelopmental disorders. So here you can see that, for example, alteration of the proliferation phase of the neural stem cells can cause microcephaly, characterized by lower brain, a smaller brain. But also the defect in the neurogenesis, in the migration of the neurons, in the generation of the neurons, or their migration, or in their connection, like including also defect during gerification, can give rise to other disorders like lissencephaly, polymicrogyria, and others. Instead, neural dysfunctions are usually associated with neuropsychiatric disorders. So here you can see different papers published by different teams showing associating functional dysfunction of the synapses or dysfunctional connection of the cortical neurons with autism spectrum disorder or schizophrenia. So late prenatal or adult neuronal and glial dysfunction are usually linked to the origin of cortical neuropsychiatric disorders, but the implication of these risk genes in the earlier phase of human brain development involving neural stem cells is not really well known. So, however, there are several papers, several works, including from Flora Maccarino's team, showing that altered hypothesizing that altered identity of the neural stem cells has earlier risk events leading to cortical dysfunctions. And also our lab recently published a paper showing association of the expression of the risk genes involved in neuropsychiatric disorders during the patterning phase and during the early radial cells development in macaque telencephalon. So here, in particular, in this work, we dissected the regions of the macaque telencephalon here from early developing macaque telencephalon, from early phase before neurogenesis up to gliogenesis. And then we performed single cell RNA sequencing. We profiled over 700,000 cells. And we could identify cells of the progenitors of the patterning centers, different radial glia cell types, or better, radial glia cells at different states of development. And of course, we could recognize the cells belonging to the excitatory or inhibitory neuronal lineage, and later gliogenic cells. This paper was recently published and once that we had is a cell atlas of the cells populating the developing monkey telencephalon. What we did was to map the expression of risk genes associated to different neuropsychiatric disorders across all the cells populating the macaque developing telencephalon. So we found that although the majority of the risk genes were enriched in the neurons, we found 132 disease-associated genes overlapping with the pattern-centered subtype markers. Of these genes, 26 were highly or exclusively expressed in the pattern-centered subtypes, and 106 risk genes were instead expressed in the pattern-centered cell at the beginning, and also later in other cells. So here you can see all it is 26 risk genes highly expressed in the pattern centers, and these are associated with neuropsychiatric disorders and are basically highly expressed in the early in these early progenitors, in the early pattern centers. So this shows that certain risk genes are expressed early in the telencephalic organizers with a pivotal role in regional pattern and neural stem cell specification. Now, of course, studying this early phase of human brain development is challenging. So, can we model human telencephalic development in vitro? Of course, we can, but we need to make sure that what we are that we are faithfully recapitulating the developmental phase that happens in vivo in vitro. So this is a work that we published a couple of years ago. This is the work that I did as postdoc with when I was postdoc with Ron Micali in Ron Micali's lab. So what we did here was we did here two types of experiments. In the first experiment, we used pluripotent stem cells, embryonic stem cells, the H9, and we differentiated the cells in neural stem cells. So we derived neural stem cells, which were using the LSB, the LSDN, the dual SMAD protocol, and then we sequentially passaged the cells, as you see here, from passage 2 to passage 8. So at each passage, we exposed the cells in parallel. We expose the cells to different phases of FGF, so different doses of FGF. So high dose of FGF induce proliferation, low dose of FGF induce differentiation. So in this case, in this way, we could reveal the differentiation potential of the neural stem cells at each phase of this progression. So this is how the stem cells look like. This is immunofluorescence for the key markers, key neural stem cell regulator, including SOX2, OTX, or PAC6. And you see that there is this progression. This is how the cells look like across the progression, right? So here you can see that the SOX2 positive and PAC6 positive, SOX2 and PAC6 positive rosettes emerge at passage 3 and passage 4, and they are not present at passage 2. So emerge at passage 3, passage 4, and then decrease across time in sequential passages like the passage 5. We perform a single bulk RNA sequencing at the end of each passage. And then RNA sequencing analysis, the RNA sequencing analysis revealed that these cells, passage 2 and passage 3 cells, these early cells express genes characteristic of the cortical hem. Instead, passage 4, neural stem cells express genes characteristic of the express genes of the cortical plate, so neuronal genes express in a more differentiated neurons like populating the cortical plate when they were exposed to low dose of FGFs. When the cells were exposed to high dose of FGF, the genes expressed were characteristic of the neurogenic radioligand cells. This analysis was done by Carlo at that time, and basically it's a COGAPS analysis. COGAPS analysis means it's a kind of a gene expression pattern analysis, identify the gene expression patterns. A little bit more sophisticated, but basically this is the concept, and a little bit more informative, but it's the concept. And then we use projection using public data set from macaque developing cortex. At the time, we didn't have the single cell RNA sequencing from our macaque paper, but we use a data set from published by Elaine team. We did another experiment, also another type of experiment, where basically we derived 6 iPS lines from 3 donors, and we differentiate now these iPS lines in neural stem cells and then directly into neurons. So we didn't apply FGF, and we didn't sequence, and we didn't pass the cells. These cells were differentiated using now adding to the LSB the dual SMAD protocol, also the inhibitor of Wnt signaling, because in this way we could avoid, we wanted to avoid to push the cells towards a dorsal telencephalic pattern, and because in this way the cells could reveal their telencephalic identity. So here again, we perform a bulk RNA sequence at different steps of this time course, and then analysis here it is a principal component analysis and a projection using human cortex data also from Elaine team. We could see that 3 of these lines here in green were biased towards dorsal telencephalic neuronal lineages. Instead, the other three lines in yellow were biased to form ventral telencephalic neuronal lineages. And interestingly, at the beginning of this time course, so at the V8, early during this differentiation progression, the dorsal bias lines express genes characteristic of the cortical hem. The ventral bias lines express genes of the anteroventral domain, anteroventral telencephalic regions, which were FGF8 and the DLK1. So here we conclude that pluripotent stem cells transition to neural stem cells recapitulate states resembling the pattern centers before traversing neural trajectory in vitro. And using this in model where basically we can control the progression of the neural stem cells in vitro, we can interrogate the early states of the early phase of human brain development, and in particular this early phase, which is the patterning, and also the early phase of neurogenesis. Now, this is we base now this new data. These are now unpublished data. This is the new work that Carlo was mentioning. We base this new data on the previously published data sets. So here, these are the COGAPS patterns ordered across the progression of these stem cells. So here you see the COGAPS enriched in genes expressed in the early passages for high and low FGF and across all the progression. We annotated these cells as organizer cells at early passages, neurogenic cells, passage 4 and passage 6, and late neurogliogenic cells, because projection of these later cells into the human macaque data showed also that these cells express, these late cells, these passage 8 cells express genes of the gliogenic genes of the gliogenic radioglial cells. Then we collected a list of risk genes associated with cortical malformations, including microcephaly, lissencephaly, etc., or neuropsychiatric disorders, including autism or schizophrenia, from other GWAS datasets. And we checked the enrichment of these genes into these COGAPS. So you see, for example, that microcephaly associated risk genes were enriched in the early passages. Lissencephalic genes were enriched in the neurogenic passages, and in the neurogenic cells here in the COGAPS enriched in these neurogenic of these neurogenic genes expressed by the passage 4, let's say. And then here we could and other diseases like ASD, for example, were enriched. These risk genes were enriched in from passage 4 to passage 8. So this shows that risk genes are expressed in neural stem cells, and neural stem cells may play a critical role in the early events leading to cortical disorders. Now, here I'm showing them the gene expression dynamics, the expression dynamics of risk genes associated to microcephaly, for example. And here you can see that the majority of these risk genes are expressed or peaks at early passages. Instead, if you take the risk genes associated with focal cortical dysplasia or enteropathies, these genes show there is a high proportion of these genes peaking at late passages, so in the late neural stem cells. It is already suggested that these two diseases are involves different types of cells. Early neural stem, different types of neural stem cells. Early neural stem cells or late neural stem cells. Then we order the different diseases based on the temporal maximum of risk genes, and we could, in this way, organize the disease as early organizers, mid-neurogenic, or late neurogliogenic disease, based on the proportion of the risk genes which are peaking at passage 2, passage 3 in the first case, passage 4 for the mid-neurogenic disease, or later or in late cells like in passage 8 neural stem cells. So here the conclusion that I can give is that there is a specific phase during neural stem cell progression that may be critical for each disease. So just to give you a quick summary of this first part, there are putative disease-specific critical phases, which are temporal. We reveal putative disease-specific critical phases, which are temporal window across human neural stem cell progression, which may be more vulnerable to dysfunctions. And there is a potential implication of the risk genes in regular cells identity specification and progression. Okay, so what we did next was to identify transcription factor networks across the human neural stem across this neural stem cell progression. So here we required that transcription factor and target were belonging to the same disease gene list and were basically co-expressed. So here we identified 31 disease regulars involving 29 core transcription factors, targeting up to 500 genes, let's say, in nine diseases. So each disease shows these regulars, these core transcription factors, which peak at a specific phase of this neural stem cell progression, in vitro neural stem cell progression, and each core transcription factor targets genes. You can see that target genes may peak also different passages than the core transcription factor. This because this indicates because the transcription factor target the transcription factor can also negatively this can reflect negative control, negative regulation of the target gene, but also the fact that one transcription factor peaks at a specific phase doesn't mean that it doesn't work to other phases. So it's less expressive but still functional. Okay, so here in this analysis we now are defining the relationship of this disease transcription factor network. So I'm showing the temporal progression of the regulars across the development of the neural stem cells. And here this analysis shows how different regulars crosstalk to each other. And this analysis is showing that there are also, showing that the different function of a transcription factor in the regulars. So a transcription factor can be a core, can be a hub transcription factor or a core transcription factor for some regular, but can be also a target for other regulars. It's also this analysis is also showing that this analysis is showing the different function of transcription factor in the regular, but also is showing how the different regulars can converge to a specific target. So this could explain why mutation associated to different diseases may show similar manifestation, but also can explain why mutation associated to the same disease may show diverse manifestation. Because if the mutation is in this transcription factor associated with ASD, for example, and it's mutated, this lesion affect early stem cells, but these other lesions this other perturbation, this transcription factor, which is expressed late, may have a different, may give me a different manifestation. And in order to see which is the effect of which is the impact of the perturbation of the different transcription factors across human corticogenesis, we perform in situ disruption simulation of the gene regulatory network using across human corticogenesis using the single cell multiomics dataset from Sergio Pasca lab. Here in this paper, in this dataset, they generated single cell multiomics across the development of the cortex of 4 different donors. We reannotated the cells because we wanted to gain resolution with the neural stem cells. So here we used the monkey paper to reannotate the more neural stem cells, and then we use the cell oracle to predict the impact of each transcription factor perturbation across radial cell progression in gliogenesis and neurogenesis. So here you can see the radial cell progression and gliogenesis trajectory. Here you can see the neurogenic trajectory. And here I'm showing them as example the knockout simulation for 2 transcription factors. KLF6, which shows a high centrality across the gliogenetic trajectory, and MEF2C, which shows high centrality in the neurogenic trajectory. So you see that green means promoted transition. Red means inhibited transition. You can see that the KLF6 knockout inhibits the progression of the radial cells, but promotes the more gliogenec phase. Instead, here there is the inhibition of the neurogenesis. This is another, here on the right, there is another representation of the same results. So the initial cells, the cells before the perturbation are on the left. The cells after the perturbation are on the right. So here you can see how the KLF6 knockout induced the perturb these early radial cells, and here how the MEF2C knockout perturb neurons, and there is accumulation of, after the perturbation accumulation, there is accumulation of the IPS cells, intermediate precursor cells, which indicate inhibited differentiation, right? Inhibited neurogenesis. And here there is the knockout simulation for each transcription factor involved in radial cells progression and gliogenesis. I want to bring your attention here on the mutation of, for example, on the perturbation of KLF4, which induce a depletion of these early radial cells. Instead, there is the perturbation of HIVP3, another transcription factor, doesn't affect the early radial cells, but affect the late radial cells. And this indicates that the transcription or that suggests that transcription factor lesions have a cell state restricted effects. So as a second summary here, I can show you that I showed you that share and disease-specific networks of cortical disorders, and the target. I showed you that share and disease-specific networks of cortical disorders, and the target genes on which multiple transcription factor lesions may converge across neural stem cell progression. Then same transcription factor perturbation impacts cells differently. There is a context-specific effect of a gene lesion, and different role. I showed you different role of transcription factor in a network, which reflects the impact of its dysfunction during neurogenesis. Now, next, what we did was to study the impact of intrinsic genetic abnormality on individual early brain development. We use always this system that I showed before, where iPS cells are differentiated into neurons and then neurons, because I say that it is recapitulate the early state of brain development. So now we want to see how the intrinsic genetic abnormality affects these early states of brain development. Of course, modeling the disease in vitro. So here we derived iPS cells from 3 control and 3 idiopathic ASD patients, and we differentiated these cells. And at day V8, the in vitro 8, so during this organizer state, we performed single-cell RNA sequencing. So we annotated the cells after integration with monkey single-cell RNA sequencing, so we could recognize that the cells in vitro were equivalent of early radial cells. They're sharing features. We found, of course, the pattern center progenitors and also later radial cells. And then we checked them by differential expression analysis. We found that the autism lines show deregulation of a patterning genes. So here there is a downregulation of genes like dorsoposterior genes like EMX2 or WLS, etc., or DMRTA1, and there is an upregulation of anteroventral genes like 4G1. It is also in line with Marianne et al. From a paper from Vaccarino's lab. So, and then we checked in individual lines what is the expression of these genes, and we found that this line, the ASD, it is ASD line, it is autistic line 384, showed upregulation of this anteroventral gene and genes and downregulation of the cortical hem genes. The other line, the 494, also shows the regulation of pattern genes, and the 375, the line 375 here in green, shows imprinting genes deregulated, imprinting genes deregulated. So then what we did was to check the expression of anteroventral organizers genes and dorsoposterior organizer genes into these lines. These genes, of course, come from the monkey datasets, and we found that the 384 line indeed express a high level of these anteroventral organizer genes and a low level of the cortical hem genes. Instead, the other line, the 494, show higher expression of more dorsal-posterior organizer like hem and zona limita infrathalamica. So we found a compromised pattern gene expression in neurostem cell from ASD affected anencephalon, and there is a putative anteroventral lineage bias of the forebrain of one individual, and a putative I would add here, putative dorsocodal lineage bias for another individual. Of course, 3 control and 3 ASD lines is very limiting, right? We need much more cells, much more lines to conclude something. But here we overcome this problem with a comparison with Jordan et al. 2023 organoid dataset. It is a paper from Vaccarino's team was published a few months ago on Nature Neuroscience. So what Alex Jordan did here was to test the differentially expressed transcription factor that we found in our 3 lines across multiple ASD lines, control ASD pairs. And we found that, in fact, these transcription factors were differentially, also resulted deregulated across the multiple lines. But in some lines were upregulated, in other ASD lines were downregulated. So what we did here was to plot the frequency of perturbation across the pairs independently of the direction, and we found that in fact this transcription factor that we found regulated in only 3 ASD lines were frequently perturbed, deregulated, also in the multiple ASD-derived neurostem cells lines. So in conclusion, I showed the potential of this in vitro modeling to explore the early phase of human brain development and the implication of early neurostem cells in brain disease. Many cortical diseases associated with these genes are expressed in the early pattern center and in neurostem cells traversing dorsal or ventral telencephalic neuronal lineages. A mutation may impact the common regulatory mechanism and affect a larger network of converging genes. So I showed you with the regular analysis that how we can explain phenotypic diversity within disorders or shared features among distinct disorders. And finally, using patient-derived neurostem cell lines, we distinguish the impact of intrinsic genetic lesions on individual early brain development, and we found recurrence of the same transcription factor alteration across multiple lines. So I want to say thank you to my PI, Pascu Rakic, and a huge thanks to the team of Gabriel Samper from Hospital del Mar Research Institute, and in particular to his PhD student, Joel Mato-Blanc, who is doing a great job on the bioinformatics related to this last unpublished work. Of course, the team of Nenad Sestan, who provide the ASD lines, Sulki Kim, JMA for helping with the single-cell RNA sequencing and the in vitro experiment, Alexandre Jardon from Vaccarino Lab, for this last analysis that I showed you across the different lines. Alvaro Duque for MacBrain resource supporting this project, and of course Carlo Colanconi from Johns Hopkins. We've been talking for years for this day about this data, and thank you to everybody for listening.
Thanks, Nico. Any questions, please?
Hi, may I just start one question? This is Novin from the very first part of your talk, you mentioned about the different comparison between different passage of neural stem cells. I just wonder if you keep them basically talking about neural stem cell, different passage of neural stem cell, you didn't differentiate them actually. The media remain FGF and EGF. Am I right for all those passages?
Yeah, you're talking about here.
Yeah, and you also have the comparison down there when you have, if you go to the slide 20 or something, I guess, or 15 perhaps, 15, 1.5.
Yeah. Yes.
So you have passage 2, 3, 4, 6, 8. We're talking about EGF, FGF here all the time.
I'm talking, right. So here, yeah. First of all, these are so these neural stem cells are there are 4 different doses of FGF, 20 nanogram per ml, 10, 0.1, and 1. So right. So basically, the 0.1 FGF condition is inducing the differentiation of the neural stem cells. But it's still, although it looks very low dose, but still there is FGF. So they are not. I mean, to differentiate the cells into neurons, I need to totally withdraw the FGF. So here I can see we can reveal differentiation potential, but still the cells are in the transition towards a specific differentiation potential, not really differentiating. It's it depends what we call differentiation or not, right? The protocol to differentiate these cells is to withdraw totally FGF abruptly. Instead, these cells were kept in a very low dose of FGF, but still, so they don't proliferate a lot, but they can show, I mean, at least transcriptomically, they show their differentiation potential.
I see your point. Thank you very much. Thank you.
Sure. You can see actually how the differentiation potential progress across the time, right? Because the point here is that these cells with the cells treated with low dose of FGF passes to express the gene of the cortical hem, right? Here I'm revealing the differentiation potential of these cells. But the cells treated with low dose of FGF in passage 4 now are basically expressing different types of genes, which are characteristic of the cortical plate. Now you don't see hem genes here; you see neuronal genes here expressed.
If I may just follow up on what you just mentioned. So have you seen any significant difference between all these 3 different concentration of, especially the highest one, which is normally we use for main NPC?
So what is the question? Sorry.
So you have the 20 microgram per ml, perhaps. That's the highest one. And when you keep it for 8 passages, when you compare it with the 0.1, I assume you're going to see a significant difference in terms of the expression of those genes that related to or associated with the neuroprogenitor cells compared to the forebrain neurons. So you're going to see less expression of the forebrain neurons in the 20 microgram. Is that correct or not necessarily?
I'm not sure I understood the question, but here basically you see the progression of these cells now express gliogenic genes. And you can, I mean, with the 20 FGF, they express genes of the gliogenic radical cells. If you treat with 0.1 FGF, you start to see now, astrocytes genes. I mean, you can see like I don't remember now, but you can see more differentiated genes expressed in the differentiated astrocytes. Let's say that.
Yeah. Got it. Thank you very much.